Nuestra siguiente presentación está a cargo de Santiago Medina Muñoz, del Angevio, y él nos va a presentar Demographic Modeling of Admixed Latin American Population from Whole Genomes. Gracias.
Hello, everyone. I'm Santiago. First, I want to thank the organizers for giving me the opportunity to present my work. And the populations in Latin America, they have a very complex demographic history with different dynamics at these distinct timescales. For example, there is the deep time history that involves the major human migrations. For instance, the out of Africa, and subsequently the peopling of the Americas. And more recently, we also have the admixture. We saw in Sarah's talk about the admixture that involved different ancestries mainly from Europe and Africa. But the problem is that there is a lack of models that accurately represent this complex history. And this is one example of a model that is available. And there is an importance in these models because in population genetics, a lot of analysis and methods rely on genetic simulations. So in order to have to test these tools, we need to have good models. And this model it has some limitations. Here we have, this is like the present, and we go back in the past. So this model here, this is an admixed population. We can think of this as Mexico. And the limitation is, for example, that it uses Asia as a proxy for Native American ancestry, which is not accurate because Native Americans have undergone further genetic differentiation. And there is also a lack of regional variation. And there is an oversimplification of admixture history. As we saw in Sarah's talks, there was also some East Asian migration into the Americas. And so we decided to develop better models for the Americas. And for that, we took data from the 1000 Genome Project and also from the MX Biobank project. Here we can see we have these continental populations, and the populations in Latin America, they are a mosaic of these ancestries. So we use data from the 1000 Genome Project, and we also use some genomes from the Biobank project that was recently published. So we also generated genomes from this project in addition to the genotype data. So first, to infer the history, first we infer the deep time history. And in this case, we infer this 4-population model that we have African, European, Asian, and the Indigenous American population. We use the joint site frequency spectrum. And now our model now it incorporates the Indigenous American populations. Before this was not possible because we didn't have Indigenous American genomes. And now with the data and with new tools, we can infer a 4-population model. And for example, we infer split time from East Asians that occur 30,000 years ago and an initial population size of 1,500 individuals. So the next step is to go to the most recent history because I told you that a mixed population has these different timescales. So for that, we use the ancestry tract length distribution, which is informative of a recent admixture history. So we first infer the local ancestry across the genome using a program called genomics. And from there, we calculate the ancestry tract length distribution that tells us about the recent admixture history. And we infer a model that encompasses the admixture dynamics. So this is like an example of the model. So here we have the present generation. Here we have the past. And this is like the ancestry proportion over time. And here we have the different migration pulses from each of the ancestries. And so we did this inference across different populations in Latin America. So now we have a model for Puerto Rico, a model for Colombia, a model for Mexico, and a model for Peru. So now we are adding to the complexity in the Americas and not just one single population, because our result shows that the migration history is very different. And now we have our 4-population model, and we add the migration history that we infer from the ancestry tracks. And we now combine this into a 5 population model that has the deep time history as well as the region and mixture dynamics. And here, this is just an example of Mexico, but this changes for each of the different regions or countries in the Americas. And this is a validation of this model. So here we have a principal component analysis. And here we can see that the principal component tree captures the variation in the Native American ancestry. The first plot here, this is from the real data. So this is here we're just plotting the PSA from the genomes. Here on the middle, we have the simulation from our model, and we can see that it mirrors the observed data. And this is the Brownian model that I showed you in the beginning. We can see that this model, because it didn't have the Native American component, it doesn't capture the variation in the Native American ancestry, which is very important for the Latin American populations. And well, finally, these models are relevant. They are important from different aspects of history. For example, the recent migrations into the Americas. And also, these models can be useful for example, for running a selection analysis. You need, when you want to run a selection scan, you sometimes need to have a null distribution. So this null distribution is going to be shaped by the demographic history. So now with a model, we can generate a null distribution and then run our selection scans. Or there are other examples, and we publish these models. They are freely available. 2 weeks ago, if you are interested, you can go and get all the models for the populations in Latin America. And with that, I want to thank my advisor, Andres Moreno, and also Aaron Raxel, my other advisor, Carmina, Ram, Diego, and Lourdes, our collaborators. And I'm happy to take questions. Yes, I'll go back to, yes, here. So for each population, we have the effective population size. So that is this present time. We also have the population sizes in the past. For example, this is like a bottleneck. So we have all those parameters. So we have also the migration rates. For example, a migration rate between Africa and Europe. So this model has the migration rates, population sizes, and split times. And it's encoded in a format called DIMS that now, like different engines, for example, Slim, MS Prime, they now are using this format that is a standard format. So with the DIMS file, you can use this model, for example, in MS Prime, in Slim, or forward PY. So that makes more easy to reuse the models. Use this part. All these other parameters, the parameters in red, they come from tracks, and the parameters in black come from the site frequency spectrum. And we just combine them.
Thanks for your talk. It's really interesting to see this kind of work. So I have a question that is related to, you said some of the genomes you used from MaxB. I wonder how many genomes you used?
Okay.
You used 1,000, yeah.
So we sequence 50 genomes. They come from the biobank. We preselected these genomes to have representation across the country. So if you plot the dots, you will see that they are spread across Mexico. And the second selection criteria was that they were more than 95% indigenous ancestry.
Okay.
And
so these are Mexican indigenous population?
Yes.
Okay. Thank you.
Hi, great talk. I just was wondering, if I understood the model correctly, you assume that the population for the admixed in Mexico was constant through time, right?
Yes, that's one of the assumptions. The effective population size is constant because with tracts you cannot infer population sizes.
Would you consider, like when the Europeans came, there was like a big bottleneck because of the scissor, right? Do you think that will change the results a lot, or do you think we don't even have to consider that?
I think it can have some effect in the results, but also is at least using, for example, the psi frequency spectrum is difficult to estimate it, because in order to estimate parameters from the allele frequencies, you need not a lot of time, but time so you can observe drift. And in the last 500 years, there is not much drift, so it gets complicated to infer like recent parameters, at least from allele frequencies. There are another methods like IBD. Mashal has been doing some work in the biobank where she inferred like effective population sizes in populations in Mexico using IBD.
Thank you so much.
Muchas gracias.